I have spent 31 years being careful. That is, above all else, what my career in paleoanthropology has been: an exercise in careful language, careful method, careful refusal to claim more than the evidence strictly permits. I have published 47 peer-reviewed papers. I have trained 19 doctoral students, several of whom have gone on to careers more distinguished than my own. I have sat on review boards and ethics committees and have been, by every institutional measure available, a responsible and rigorous scientist. I am telling you this so that you understand the weight of what I'm about to say. The Yeti is not an undiscovered great ape. It is not a relic population of Gigantopithecus blacki. It is not a misidentified bear, not a cultural myth, not the product of altitude sickness and wishful thinking. The Yeti is something that the available framework of evolutionary biology does not have a clean category for. And the reason it does not have a clean category is that its origin, its actual evidenced, documented origin, which I have spent the last 11 years of my career assembling from sources across 4 scientific disciplines, rewrites a portion of human prehistory that we had believed until very recently was settled. The Yeti is, in the most precise and unsettling sense I know how to deploy those words, partially us. I have been trying to find a way to publish this finding through conventional channels for 6 years. I have submitted to 3 journals. I have been rejected twice on methodological grounds that I believe and can demonstrate are pretextual, and once with a reviewer comment that said, with a directness I actually respected, the implications of this paper, if the data holds, are too significant to publish without institutional consensus that does not yet exist. That reviewer was correct. The consensus does not exist. I am publishing here in this form because the alternative is to die with the data in a filing cabinet, and I have decided that is not acceptable. My name is Dr. Henrik Solberg. I am 63 years old. I was born in Bergen, Norway, and I have spent the working decades of my life studying the genetic and archaeological evidence of archaic human populations in High Asia. What follows is the account of how I arrived at a conclusion that will, I believe, eventually be recognized as correct, and the story of what that journey cost me and what it revealed. My research revealed something profound that I did not anticipate when I first encountered it in 2008 in the form of a 40,000-year-old finger bone from a cave in the Altai Mountains of Siberia, the Denisova cave specimen, which would eventually give its name to the Denisovans, a previously unknown archaic human population, was not my primary research focus at the time. I was working on a separate project involving mitochondrial DNA extraction from late Pleistocene sediment samples in the Tibetan Plateau region. But the Denisovan results, when they were published by the Max Planck Institute in 2010, intersected with my own data in a way I could not dismiss. The Denisovans, the genetic analysis showed, had interbred with anatomically modern humans. The evidence of this interbreeding, Denisovan DNA sequences present in the genomes of contemporary populations across South and Southeast Asia, with the highest concentrations in Melanesian and Tibetan populations, was not controversial once the data was available. It confirmed what population geneticists had been arguing theoretically for years, that the story of modern human origins was not a clean replacement narrative, but a complex multi-episode mixing of populations across both time and geography. What made me sit very still at my desk in the Uppsala University Genetics Facility on March 14, 2011, was a specific data point in my own sediment analysis. I had been extracting and sequencing ancient DNA from permafrost cores taken at high altitude in the Tibetan Plateau, looking for evidence of Denisovan occupation of the region. What I found in a core from approximately 38,000 years before present was a sequence I could not place. It matched no known archaic human population. It was not Homo sapiens, it was not Neanderthal, it was not Denisovan, though it shared certain marker sequences with the Denisovan genome in proportions that suggested a relationship. It was, by every analytical tool available to me, something adjacent to the Denisovan lineage, but distinct from it. A branch point I had no prior reference for. I labeled it Hominid X in my working notes. I ran the analysis 4 times across 2 separate samples. The sequence was consistent and reproducible. I did not publish it. I needed more data. What I did was begin, quietly and with the specific obsessive patience that characterizes the best and worst impulses of research science, to look for more. Here's why this changed everything. For me, in 2013, a paper was published in the journal Proceedings of the Royal Society B by Dr. Brian Sykes of Oxford University, analyzing the mitochondrial DNA of hair samples attributed to the Yeti and the Almas across multiple Himalayan and Central Asian sources. The paper was rigorous, the methodology sound, and the conclusions carefully framed. Most of the samples matched known species as expected. But 2 samples, one from the Himalayas, one from Ladakh, matched an ancient polar bear sequence that implied a hybrid population of uncertain status. The media reported this as Yeti solved. It's a bear. This was not what the paper said, and anyone who read it carefully knew it was not what the paper said. What the paper said was that 2 samples matched a sequence associated with an ancient hybridization event, and that the implications required further investigation. I contacted Dr. Sykes and requested access to the raw sequence data for those 2 samples. He was generous and prompt. I ran my own comparative analysis against the Hominid X sequence from my 2011 sediment extraction. The match was partial, not complete. The mitochondrial sequences from the hair samples were not identical to my sediment sequence, but the degree of overlap in specific marker regions that I had not published and that Sykes's team could not have known to look for was statistically impossible to explain by chance. The probability of that degree of alignment occurring randomly, given the sample sizes and sequence lengths involved, was less than 1 in 800,000. I sat with that number for a long time. I am a careful man. I do not rush to conclusions. I have built a career on the discipline of not rushing to conclusions. Then I began planning the expedition. Before I could plan the expedition, I needed to know where to look. The genomic data told me what I was looking for. The oral record, which I spent 3 years systematically documenting between 2012 and 2015, told me where. I want to be precise about my methodology here because the oral tradition work is the part of this project most likely to be dismissed by colleagues who have not done it themselves. The collection and analysis of indigenous oral knowledge is a legitimate and rigorous scientific method when it is applied with the same standards of consistency, cross-referencing, and critical evaluation that any primary source material demands. I applied those standards. The oral record did not fail them. My field research coordinator, Tenzin, was the essential figure in this phase. He had family connections across 3 of the major Sherpa communities in the Khumbu and Rolwailing regions, and earned relationships with Rai and Limbu communities in the eastern hill districts that went back 15 years of shared fieldwork. He understood what I was looking for, and he understood, crucially, how to ask for it in ways that the communities would receive as respectful inquiry rather than extraction. The protocol we developed was straightforward: extended residence in each community, participation in daily life, explicit transparency about the research purpose, and documentation only with the knowledge and consent of the participants. We were not collecting folklore. We were documenting observational knowledge that had been maintained and transmitted across generations by people who had direct and continuous experience of the terrain we were studying. Over 3 years and 14 communities, we documented 237 separate accounts that met our inclusion criteria for specificity, internal consistency, and independence from media-derived Yeti imagery. The inclusion criteria were strict. Accounts had to reference specific locations by local topographic names, specific seasonal patterns, specific behavioral observations that were consistent across multiple independent informants, and had to be transmitted through family or community lines rather than derived from secondary sources. The geographic clustering that emerged from this analysis was striking. 83% of the qualifying accounts clustered in 7 distinct high-altitude zones, all above 4,200 meters, all in terrain that shares specific geological and ecological characteristics. South-facing rock shelter formations with reliable water sources, proximity to high-altitude ungulate migration routes, and access to the upper vegetation zone that provides the plant material documented in the diet-related accounts. 3 of these 7 zones, the ones with the densest and most temporally deep account clustering, became the primary targets for the 2015 expedition. The Kanchenjunga Conservation Area Zone was selected first because it had the highest density of qualifying accounts, the deepest temporal depth in the oral record, and the most specific geographic references. Tenzin had 3 separate family connections with communities in the buffer zone whose knowledge of the specific rock shelter we eventually sampled went back at least 4 generations of direct observation. What most people don't understand about oral tradition as scientific data is that its reliability is not a function of how old it is, but of how consistently it has been transmitted and tested against direct observation. A community that has been living alongside a population for generations and has direct, ongoing experience of that population's presence develops an observational record that is in important respects more reliable than sporadic Western encounter accounts, because it has been continuously corrected by experience rather than frozen in a single moment of shock or confusion. The communities whose knowledge guided our expedition were not telling us stories. They were sharing a field record that had been refined across generations of direct observation. When we found the sequence in the shelter sediment, I was not surprised. I was confirming what that record had already told me. I did not tell my department chair what I was looking for. I described the project as a continuation of my plateau sediment work, which was true, and I applied for funding through a private foundation that had supported my previous High Asia research, which I received. I assembled a small team, 2 graduate students who knew the sediment work and not the deeper agenda, a Nepali research coordinator named Tenzin, who had been indispensable to my previous fieldwork and who I trusted with my life, and eventually with considerably more, and a specialist in environmental DNA extraction from the University of Copenhagen named Dr. Astrid Vang, who was the only other person I told the full scope of the hypothesis to before we left. Dr. Vang's response to the full briefing, delivered in a coffee shop near the Copenhagen airport on September 3, 2015, was to sit quietly for a full minute and then say, "If you're right, every textbook I have helped right is wrong in at least one chapter." Then she said, "When do we leave?" She is, in my experience, the best kind of scientist. The kind who is more interested in what is true than in what is convenient. The Kanchenjunga Conservation Area sits at the junction of Nepal, Sikkim, and Tibet. It is one of the least studied high-altitude ecosystems in the Himalayas, in part because of its political complexity, and in part because the terrain above 5,000 meters is genuinely hostile to extended research presence. We established a base camp at 4,200 meters and spent the first 2 weeks conducting the sediment survey that was our official purpose. What we were actually doing in the margins of that work was collecting environmental DNA samples from specific locations identified in the oral accounts I had spent 3 years gathering from Sherpa, Rai, and Limbu communities across the region. The oral accounts, which I had treated initially as cultural data and gradually come to treat as something closer to observational reports, consistently described the same locations for Yeti activity across multiple generations and multiple communities. The consistency was itself data. Oral traditions do not maintain geographic specificity across generations unless the geographic specificity corresponds to something real. On October 19, 2015, Dr. Vang extracted environmental DNA from a rock shelter at 4,600 meters that 3 separate community accounts had identified as a site of regular Yeti presence within living memory. The shelter was not a cave exactly, a deep overhang, south-facing, with evidence of long-term occupancy visible in the floor sediment, ash deposits, processed bone fragments, a section of floor that had been deliberately leveled. The eDNA results came back from the Copenhagen lab 6 weeks after our return. The shelter contained sequences matching Homo sapiens as expected, possibly from our own team, possibly historical. It contained sequences from several known high-altitude species, and it contained in 4 separate samples from different sections of the floor sediment at different depths. The hominid X sequence, not a partial match this time, a full match across all marker regions with 97.3% confidence. Dr. Vang called me from Copenhagen at 11 in the evening. She said, "Henrik, it's in the shelter. It's definitely in the shelter." Then neither of us spoke for a while. The genetic analysis I'm about to describe represents 4 years of work conducted across 3 laboratory facilities involving 9 researchers at various stages of the process, most of whom did not have the full context of what they were analyzing. I compartmentalized deliberately, not out of secrecy for its own sake, but because I had learned from the partial disclosure I made to a senior colleague in 2016 that I will describe shortly, what full early disclosure would produce. The hominid X genome, assembled from the shelter eDNA, The sediment sequences and 2 additional physical samples I will account for later is a mosaic. This is the word I keep returning to because it is the most accurate. It contains approximately 62% sequences consistent with late archaic Denisovan populations, specifically the eastern Denisovan clade whose range appears to have extended through the Tibetan Plateau and into the Himalayan Massif. This component is the oldest phylogenetically and represents the deepest ancestral layer. Approximately 23% sequences consistent with early anatomically modern Homo sapiens, specifically the South Asian dispersal event that genetic studies place at between 40 and 60,000 years before present. This component shows the specific mutation patterns associated with high-altitude adaptation, including the EPAS1 gene variant that provides the exceptional oxygen processing efficiency documented in contemporary Tibetan populations. Significantly, the hominid X version of this variant is more extreme than any documented in living humans. Approximately 15% sequences that match neither known archaic human population nor any other sequenced hominin. This component is the one that keeps me awake. It is old. Phylogenetic dating suggests a divergence from the main human lineage of somewhere between 300,000 and 500,000 years before present. It is not Homo heidelbergensis, and it is not Homo naledi, and it does not match any of the several partial archaic genomes currently in the databases. It is something we have not named yet, because we do not have enough of it to name confidently. What this mosaic describes in population genetic terms is a hybridization event, or more likely, a series of hybridization events across an extended period between at least 3 distinct hominin populations in the High Asian region, occurring roughly 40 to 60,000 years before present, during the period when modern humans were first dispersing into the region and encountering its existing archaic inhabitants. My research revealed something profound that took me 2 years to fully accept. What we call the Yeti is the living descendant of that hybridization, not a great ape, not a bear, a hominin, an archaic human population shaped by the most extreme high-altitude environment on the planet that has persisted into the present in a form that our taxonomy has no existing category for. We share, depending on how you count the overlapping sequences, between 23 and 31% of our genome with this population. They are not our ancestors, but they are, unambiguously, our relatives. In November 2016, before the full genomic picture was assembled, I made a partial disclosure to a colleague I will not name who held a senior position at a major European research institution and whose opinion I valued and whose discretion I trusted incorrectly. I told him the core finding that I had identified a novel hominin sequence in high Asian environmental samples and hair analysis that appeared to represent a living population with a Denisovan-adjacent origin. I showed him the preliminary alignment data. I explained the confidence levels. He was quiet for a long time. Then he said, "Do you understand what happens to your career if you publish this without ironclad consensus support?" I said I understood the risks. He said, "You don't? You think you do? You don't." He did not betray my confidence directly. What he did was begin quietly and through the specific mechanisms that senior figures in academic institutions have available to them to create conditions in which the funding for my continued High Asia research became progressively more difficult to secure. 2 grants I had strong records for were declined in the following 18 months, in both cases with reviewer comments that referenced methodological concerns I had addressed in my submissions and that I believe were generated without close reading of the actual applications. What most people don't understand about academic science is that the suppression of inconvenient findings rarely requires conspiracy. It requires only the quiet exercise of institutional gatekeeping by people who have concluded, in good faith or otherwise, that the finding is too disruptive to allow. The colleague who undermined my funding believed, I think genuinely, that he was protecting me from myself. The reviewers who declined my grants may have believed they were maintaining methodological standards. The result was the same. The work was slowed, the publication timeline extended, and I spent 18 months reestablishing the funding base from private sources that were willing to support the research without institutional oversight. I am not bitter about this. I am reporting it because it is part of the record and because the record should be complete. I need to be precise about the physical samples because they are the most sensitive part of this account and the part most subject to misrepresentation. Between 2016 and 2019, through a combination of my own field work and the remarkable networks of the regional communities whose oral knowledge had guided the investigation from the beginning, I obtained 2 physical biological samples that I am confident are attributable to the living population I am describing. The first was a hair sample obtained in January 2017 from a location in the Makalu Barun National Park at approximately 5,100 meters, provided by a Rai community elder who had been a part of the broader oral documentation project and who understood, in ways I had spent 3 years earning, that I was conducting serious science and not hunting for trophies or tabloid material. The sample was 12 centimeters in length, reddish-gray in color, with a follicle intact. I transported it under chain of custody protocols to the Copenhagen lab. The nuclear DNA extraction from this sample took 4 months. Nuclear DNA from hair is significantly more challenging to extract and sequence than mitochondrial DNA, and the sample degradation from the field collection conditions was considerable. What we obtained was partial but sufficient. Coverage of approximately 30% of the nuclear genome in the regions of highest confidence, matching the hominid X sequence with 96.1% concordance. The second sample was collected by Tenzin, my research coordinator, in August 2018 from a site in the Kangchenjunga buffer zone. It was a fragment of integument, shed skin most likely, adhering to a section of rock at a location Tenzin identified as a regular passage site based on his own long-term observations. This sample yielded nuclear DNA of higher quality than the hair, with coverage of approximately 44% of the genome. The 2 nuclear sequences are consistent with each other and with the hominid X mosaic I have described. They are also consistent with the environmental DNA from the shelter. Across 4 independent data sources, in samples obtained at different times and locations and processed in different laboratory facilities, the sequence is coherent and reproducible. This is, by the standards of the field, sufficient evidence to describe a new taxon. The scientific name I have proposed in my unpublished manuscript is Homo altaiensis himalayensis, a designation that places the population within the genus Homo, based on its genomic relationship to other members of that genus. while marking its geographic origin and its specific adaptive lineage. I expect this designation to be contested. I expect the entire paper to be contested. I am publishing it anyway. Here is where the science and the encounter accounts converge, and where I think the convergence is the most important part of the story. The Homo altaiensis, Himalayensis genome contains several functional gene regions that, when compared to known human variants, suggest specific physiological and neurological characteristics. I want to be careful about the limits of what genomic analysis can claim. Genotype does not determine phenotype with the simplicity that popular science communication often implies. But I also want to be direct about what the data implies because the implications are significant. The EPAS1 variant in the hominid X genome, as I noted, exceeds the efficiency of the most extreme human version documented in Tibetan populations. The modeling suggests an organism capable of maintaining normal cognitive and physical function at oxygen partial pressures that would produce incapacitation in an acclimatized climber. This is consistent with the behavioral evidence from encounter accounts. Yeti activity concentrated at elevations that are at or above the physiological ceiling for sustained human presence. The neurological gene regions are the most striking. Several variants in the prefrontal cortex development pathway, the brain regions most associated with executive function, social cognition, and what we broadly call theory of mind, the capacity to model the mental states of others, show a pattern I did not anticipate. They are not reduced relative to Homo sapiens. They are, in 3 specific regions, expanded. My observations consistently showed in cross-referencing the genomic data with the behavioral literature that the most reliable and consistent feature of Yeti encounter accounts across cultures and centuries is not the creature's size or its fearsome appearance, but its observational behavior, the specific quality of attention it brings to the humans it encounters, characterized by calculation, assessment, and what witnesses consistently describe as the disconcerting impression of being understood rather than merely detected. The genomic data does not prove this. It is entirely consistent with it. The social cognition finding has a second implication that I find more difficult to sit with. Theory of mind, the capacity to model other minds, is the cognitive foundation of language, culture, and the kind of intergenerational knowledge transmission that produces what we call civilization. The expanded variants in these regions in the hominid X genome suggest a population capable of exactly this kind of transmission, not in the form we recognize, not in written language or material culture in the conventional sense, but in forms appropriate to a small, highly mobile, largely nocturnal population living in terrain that punishes any form of fixed settlement. Oral transmission, territorial knowledge passed across generations, the kind of deep ecological expertise that the most credible encounter accounts, particularly those from the communities who have lived alongside this population for centuries, consistently attribute to them. They are not primitive. They are differently adapted. The distinction matters enormously. The genomic story became fully coherent for me only when I integrated it with archaeological data that had been, until this point in my career, outside my primary expertise. In 2019, I spent 3 months in collaboration with an archaeologist colleague at the University of Kathmandu, Dr. Purnima Rai, whose work on Mesolithic and Neolithic occupation patterns in the Himalayan foothills is meticulous, and whose willingness to extend her analytical framework to include the implications of my genetic data was, I should say plainly, professionally courageous. Reviewing the site record for high-altitude locations above 4,000 meters across the Nepal Himalaya. What most people don't understand is that the high-altitude archaeological record in this region is extraordinarily incomplete. Excavation at extreme altitude is expensive, physically demanding, and institutionally under-prioritized. The known sites are few, the dating often imprecise, and the interpretive framework has been built almost entirely around the assumption that any evidence of human activity above 4,000 meters represents either seasonal herding or expedition-era presence. Dr. Rai and I identified 14 sites in the existing literature whose evidence did not fit this framework. Sites with ash and processed bone at elevations that exceed the permanent herding altitude by more than 500 meters. Sites with evidence of long-term repeated occupation that predates any documented herding culture in the region by thousands of years. Sites with stone tool assemblages that do not match any known Mesolithic or Neolithic tradition from the broader region, but that show consistent internal logic and consistent temporal depth. At 3 of these sites, with Dr. Rai's team conducting the fieldwork, we obtained sediment samples for eDNA analysis. At 2 of the 3, the hominid X sequence was present. The oldest confirmed hominid X eDNA from these sites dates to approximately 23,000 years before present. This places the population in the Himalayan Massif during the last glacial maximum, the period when ice sheets covered much of the northern hemisphere and human populations in most of the world were retreating to lower altitude refugia. The hominid X population was not retreating. It was, apparently, resident. High altitude during the glacial maximum was not a refuge for them. It was home. I have thought about what 23,000 years of continuous habitation of the highest terrain on the planet means for the adaptive biology of this population. And I keep arriving at the same conclusion. We are not looking at survivors of a retreating lineage. We are looking at a population that has been optimizing for this specific environment for longer than modern humans have been on this continent at all. I have debated whether to include this account, because it is the most personal section of this testimony and the one most likely to undermine my credibility with readers who have been willing to follow the genomic and archaeological argument this far. I have decided to include it because the record should be complete, and because omitting the most direct evidence I have in the interest of appearing more scientific would be a form of dishonesty I cannot endorse. On August 22, 2018, at approximately 2 in the morning, on the ridge above the Kangchenjunga Buffer Zone camp, where Tenzin and I were conducting the second phase of the eDNA survey, I had a direct encounter with a member of the population I have been describing. I was outside the tent. I had been unable to sleep. I am a poor sleeper at altitude in the best circumstances, and the previous day's sample collection had produced preliminary field readings that I had been processing for hours. And I had gone out to walk the camp perimeter in the clear, cold air. The moon was past full, but still bright enough to read by. The terrain above the camp was a section of rocky moraine leading to the upper snowfield. The creature was on the moraine approximately 120 meters above me. I saw it because it moved. a single, deliberate movement from one rock formation to another that resolved, as my eyes adjusted, into a shape I could not attribute to any natural feature of the terrain. I stood very still. I have been in the field long enough to know that the impulse to move is almost always wrong. It was large. At that distance and in that light, I could not make reliable estimates with the confidence I would want for a published account. So I will say only large, in the way that the encounter descriptions consistently say, which is large beyond the immediate register of the human visual system's calibration for large. The silhouette against the snow was bipedal and upright, and held that posture with the effortless steadiness of something that has been standing on this terrain in these conditions its entire life. It was looking at me. I want to be precise about what I mean by this. At 120 meters in moonlight, I could not see its eyes. I could not see its face with any resolution. What I mean is that its entire physical orientation was toward me with a quality of fixed attention that I have spent 30 years observing in field animals, and that I have never seen expressed with this degree of stillness and duration. It was not the scanning attention of an animal surveilling its environment. It was the focused, unmoving attention of something that had already identified me and was now studying me. We were in that configuration for, by my subsequent estimate, between 6 and 9 minutes. I did not move. It did not move. The night was completely still. Then it turned and walked up the moraine toward the snowfield, and within 30 seconds was indistinguishable from the landscape, and I was standing alone in the cold, with my field notebook in my hand and the specific quality of stillness that follows a genuinely significant event. I went back into the tent and wrote 4 pages of notes, timed from 2:17 to 2:51 a.m. The notes are in Journal 23. I described the physical characteristics as precisely as the conditions allowed, the duration and quality of the encounter, my own emotional and physiological state during it, and one observation that I have returned to more than any other in the years since. During the entire 6 to 9 minutes of the encounter, I felt no threat. Not the managed absence of threat that you feel when you are in the presence of a large animal, and reminding yourself not to panic. The genuine absence of it. The creature's attention was intense and sustained and completely directed at me, and it produced in me, after the first minute, not fear, but something I can only describe as the feeling of being genuinely seen by a genuinely attentive intelligence. This is, I am aware, consistent with the expanded social cognition variants I would document in the genome 6 months later from Tenzin's integument sample. It is also consistent with every credible encounter account I have read. The witnesses were not misidentifying bears or projecting meaning onto animal blankness. They were describing something real. I know this now in the way that only direct experience allows you to know it, and the direct experience confirms what the genomic analysis had already told me. This population is present, it is attending, and it is considerably more aware of us than we have been of it. I want to spend some time on the encounter accounts, because they are what most people engage with first when they think about the Yeti, and because reading them through the lens of what I now know about the population's biology and history produces an experience I can only describe as vertiginous. I have read, in the course of this research, several hundred documented encounter accounts spanning 3 centuries, drawn from sources across Nepal, Tibet, Bhutan, Sikkim, and the broader Himalayan region. I have read them in translation, in original, where my language is allowed, and with the systematic attention I bring to any primary source material. The consistency is striking in ways that the skeptical literature has always acknowledged without, I believe, drawing the right conclusions from. Skeptics point to consistency as evidence of cultural transmission, that accounts are consistent because people are repeating a shared cultural template rather than independently reporting observations. This argument has some validity for the most dramatic and symbol-laden accounts. It does not adequately explain the consistency of specific behavioral details that have no obvious cultural valence. The observational stillness, the preference for specific elevation bands, the avoidance of fire but not of its warmth from a distance, the consistent reports of what witnesses describe as deliberate concealment behavior that goes beyond simple flight. What strikes me most forcefully, reading the accounts now, is the consistent characterization of the creature's attention as reciprocal. Witnesses do not describe being detected by a predator or startled by an animal. They describe being studied by something that is as interested in understanding them as they are, in their retrospective accounts, in understanding it. This is the specific quality of attention that the expanded social cognition variants in the hominid X genome predict. The community accounts, particularly from the Sherpa, Rai, and Limbu communities whose oral traditions have the most developed and contextually specific framework for the Yeti, consistently describe it not as a monster, but as a neighbor. A difficult, unpredictable, sometimes dangerous neighbor, but a neighbor. A presence that shares the landscape, that has its own purposes and its own history, that is understood through long experience to be navigable if approached with appropriate knowledge and respect. This framing makes complete sense for a community that has been coexisting with an archaic hominin population for generations. It makes very little sense as a cultural fantasy about a monster. I am a scientist. The implications of a finding are not my primary responsibility. The finding itself is my responsibility and the rigor of the method that produced it. But I am also a human being, and the implications of this finding have occupied a significant portion of my thinking for the last several years. And I think I owe you an honest account of what that occupation looks like. The first implication is taxonomic and, on its face, dry. We have a new member of the genus Homo. This will require a revision of the standard account of human evolution in High Asia, an expansion of the known geographic range of Denisovan hybridization, and the retirement of several hypotheses about the Yeti's biological identity that have occupied the field for decades. This is significant but manageable. The second implication is not dry at all. This population is living, not fossil, not ancient, living in the present in terrain that has protected it from the documentation it has otherwise been unable to escape in the encounter record. There are living members of the genus Homo that are not Homo sapiens, that are related to us but not us, adapted to an environment we can barely survive, carrying a genome that includes our own sequences alongside sequences we have not yet fully characterized. What do we owe them? I am not a philosopher or an ethicist, and I am aware that I am not equipped to answer that question definitively. But I note that the legal and ethical frameworks governing the rights of living creatures are built on taxonomic categories, and that a living member of the genus Homo falls into a category that our current frameworks have not been designed to address. The closest precedent. The debate about personhood and rights in relation to great apes, which has been ongoing for 40 years without resolution, is the closest precedent and is also entirely inadequate. The third implication is the one I find myself returning to most often, because it is the one with the deepest roots in the work itself. The hominid X population has been resident in the Himalayan Massif for at least 23,000 years. It has survived the last glacial maximum, the Holocene transition, the agricultural revolution, the development of trade routes through the Himalayan passes, the arrival of mountaineering expeditions in the 20th century, and the subsequent explosion of high-altitude human activity that has fundamentally changed the character of the environment above 5,000 meters. It has survived all of this through a combination of extreme physiological adaptation, behavioral intelligence, and the specific advantage of occupying terrain that remained, until this last century, practically inaccessible to the expanding world below. That advantage is diminishing. The retreat elevation increases every decade as human activity follows the infrastructure upward. The population whose existence I am describing has a smaller viable range today than it had 50 years ago, and a smaller range 50 years ago than it had a century before that. I do not know the current population size. I have estimates based on the eDNA density data that suggest a small but not yet critically reduced number. I have no certainty. What I have is the data, and the data says this is a population that deserves the same consideration we give to any other endangered member of the genus Homo. The fact that we did not know it existed does not relieve us of the obligation that its existence creates. I have been asked by the small number of people who know the scope of this research why I am publishing it in this form rather than waiting for the institutional consensus that would allow a conventional peer-reviewed publication. The question deserves a direct answer. The institutional consensus is not coming. I have understood this since 2019, when the third journal submission was declined with the reviewer comment I described in the opening of this account. The finding is too large for the normal channels to process, and the normal channels will not accelerate their processing because the finding requires it. Institutions do not work that way. What I can do, what I am doing, is create a public record, make the finding visible in a form that cannot be quietly declined and filed away. Invite the scrutiny that will come, because the scrutiny is necessary and I welcome it. Challenge other scientists with the data to replicate, extend, or refute it through their own work. I am also, and I want to be honest about this, 73 years old. No, 63, I age myself in my own account. And I have been working on this for 31 years, and there is a version of my death in which the filing cabinets contain everything, and the finding dies with me. And I have decided, with increasing conviction over the last 2 years, that this would be a failure of my responsibility to the work and to the population it concerns. My research revealed something profound about the nature of scientific courage that I did not expect to learn at 63. The most important act of rigor is sometimes publishing before you are ready, rather than waiting until the institution tells you that you are. Readiness, in the conventional sense, would require a consensus that the finding itself is structuring the institutions against. Waiting for that consensus is not caution. It is capitulation. I have the data. I have the methodology. I have 31 years of careful work and 4 independent data sources and a genomic analysis that I can defend line by line against any scrutiny that engages it honestly. I am publishing. I want to close with what I think this means, not for my career or for the taxonomy of Himalayan hominins or for the journals that have declined my submissions, but for the broader question of what we are and what we share this planet with. For most of recorded history, Homo sapiens has operated on the assumption that we are alone. Not alone in the cosmos, necessarily. That debate is older than science, but alone on this planet as members of the genus Homo, the last of our kind, the final survivors of what was, during the Middle and Late Pleistocene, a relatively crowded field of hominin populations occupying overlapping ranges and interbreeding at their margins and competing for the resources of a planet that neither they nor we had yet fundamentally altered. We are not alone. We have not been alone. There is a population of our relatives living in the highest mountains on Earth, carrying our shared evolutionary history in their genome alongside sequences from lineages we are only beginning to characterize, adapted to an environment that we can barely survive, watching us with the specific attention of a mind built for exactly this kind of watching, as we have expanded into the spaces they still occupy. I think about what it means to have been watched across the full span of human recorded history by something that shares our genus and exceeds our altitude and has its own understanding of what we are and what we represent to the terrain they have inhabited for 23,000 years. I think about the specific quality of attention that every credible encounter account describes. The stillness, the calculation, the impression of being understood. I think about the expanded social cognition variants in the genome that predict exactly this quality of attention. And I think about what it would mean to be that population, to have been that population, watching the expanding footprint of your nearest relative climb higher and higher up the mountain every generation. The Yeti is not a monster. It is not a myth. It is not a misidentified bear. It is a member of our genus, our relative, our contemporary, our elder in this specific landscape by a margin of tens of thousands of years. It is the last surviving evidence that the human story in High Asia is older and stranger and more populated than we had believed, and that the loneliness we assumed was ours alone is perhaps a condition we share. What most people don't understand is that the discovery of the Yeti's origin is not a discovery about the Yeti. It is a discovery about us, about what we are part of, what we have forgotten, and what has been patiently, quietly, from an elevation we can barely reach, waiting for us to remember. I am 63 years old. I have spent 31 years on this question. I believe I have found the answer, and I believe the answer will eventually be recognized as correct. And I believe that recognition will change something fundamental in how we understand our place in this world. That is enough. That is finally enough.